Hi guys, and welcome to our module on Gametogenesis. Now, this particular module is under the introductory section of embryology, and the reason why it's important for us to talk about gametogenesis before diving into embryology proper is because of the fact that the starting point of embryology, which is fertilization, stems from the union between two major s- forms of special cells, and these cells are known as gametes. So gametes are specialized body cells that are haploid in nature and that are involved in the process of fertilization. And the production of these cells usually is what we know as gametogenesis. Gametogenesis is described as the process by which the specialized generative cells of gametes, the sperm cells in males and the oocyte in females, are formed and developed. For gametogenesis to occur, either in males or females, it tends to undergo two major processes. The first is usually meiosis. Meiosis involves the reduction of a diploid form of cell to the haploid form of cell. Then finally, we have the gamete maturation, in which the gametes would now mature further to become either a sperm cell or an oocyte. So about 99% of cells found in our body are diploid in nature, and this includes cells that are found within the specialized structures involved in gametogenesis, which would be the testes in males and the ovaries in females. But a few chunk of these cells tend to undergo a special process known as meiosis, and meiosis practically involves the conversion of that diploid form of cell to the haploid form. And the haploid form will now further mature through the process of gamete maturation to give rise to a fully mature gamete. Now let's talk about meiosis in this sense. So meiosis is the very first aspect of gametogenesis. It is the conversion of the diploid cells to haploid cells. What the diploid cell is a cell that contains a total of 46 chromosomes, or we call 23 N chromosomes. That's 23 from the mother side and 23 from the father side. So every single cell in the body has a total of 46 chromosomes. But from when through meiosis, we have this 46 set of chromosomes present in a single cell being reduced to a total of 23 set of chromosomes, which is what we call a haploid cell. So meiosis is simply the specialized process by which this takes place. And as shown in the image below, you'll notice that meiosis usually tends to be divided into two phases. We have meiosis 1 and meiosis 2. And at the end of the day, we go from having 23 N chromosomes or 46 chromosomes to a total of 23 chromosomes as shown in the image below. This form of reaction or this form of cell division is known as a reduction reaction, and that is in contrast to mitosis. Mitosis is known as a duplication reaction because we go from for the six set of chromosomes in the adult or parent cell to the same for the sixth set of chromosomes in the daughter cell. But in the case of meiosis, it's known as a reduction reaction because we go from for the sixth set of chromosomes in the adult cell to 23 set of chromosomes in the daughter cell. So let's talk about the different stages of meiosis. We start with meiosis 1. Meiosis 1 is divided into four major subphases, which includes the prophase 1, metaphase 1, anaphase 1, and telophase 1. And then finally, from there, we move on to meiosis 2. We'll talk about meiosis 2. So let's start with meiosis 1. In meiosis 1, we have the very first subphase of meiosis 1 known as prophase. And that's usually typically follows a similar fashion to what occurs in mitosis. Mitosis also has its own set of subphases: prophase, metaphase, anaphase, and telophase. So what happens during prophase 1 is similar to what happens during prophase in mitosis. And what does that entail? So it entails, first of all, the primary gametocytes being present with 46 duplicated chromosomes. So now we go from 46 to about 92 chromosomes around prophase 1. And this is important to note because we start off with 46 chromosomes, but during the cell cycle, around the S phase of cell cycle, that 46 chromosomes is duplicated into a total of 92 chromosomes. So at the start of meiosis 1, we actually have a total of 46 duplicated chromosomes, or 4n, a total of 92 chromosomes starting it. And this chromosome starts to condense, and as they condense, they become more visible. With time, we have the centrosome also separating, we have the mitotic spindles forming, and we have the nuclear membrane disintegrating. These are things that we talked about when we talked about the process of mitosis. Aside for these unique features that occurs that is similar to what we talked about in mitosis, two important things happen in prophase one that we don't see in mitosis. So the first is what we call a synapsis or pairing of homologous duplicated chromosomes. So remember that in prior to meiosis one, we had a total of 23 N chromosomes. That N means 23 times 2, which is 46 chromosomes. 23 of these came from the father, 23 came from the mother. So 23 of the chromosomes is from the father, 23 is from the mother. And like I said, before prophase 1 begins, we have this now further being duplicated into a total of 4N chromosomes. So we go from having 46 chromosomes as 23 mother and 23 at the father side to having a total of 46 chromosomes on the mother side and 46 chromosomes on the father side, making a total of 92 chromosomes. And like I said, this is what we start with. Once we get to prophase 1, each of the chromosomes that are paired that are either from the mother side or father side start to pair with one another. So let's say chromosome 1 from the mother side will pair up with chromosome 1 from the father side. Out of those 46 chromosomes, chromosome 2 in the mother side will pair up with chromosome 2 in the father side, and like that we have this pairing occurring. So that pairing is what we call synapsis, and it is homologous, meaning that we have the chromosome that is unique or is identical in the mother side will always pair with that in the father side. That's why it is homologous. We don't have random pairing occurring. So once this occurs, once this pairing or synapsis occurs, the second important thing that happens is that there is crossing over and exchange of genetic materials between the paired homologous chromosome. So if we have a gene, let's say for example, in chromosome 1, let's say this is chromosome 1 from the mother side, let's say we have the gene, and the gene in this particular chromosome is maybe the gene for height. In this particular chromosome, the gene is a gene that would have a phenotype of short height, and that gene, that particular chromosome has been paired up with the homologous fashion or homologous chromosome from the father side. So let's say chromosome 1 from the father side. That chromosome 1 also has its own set of genes. So let's say that one has a tall gene on that father side. Once synapses occurs and the crossing over occurs, what happens is that a chunk of the tall gene can be shifted or exchanged for that of the short gene. So you can have the tall gene going to the mother's chromosome 1 and the short gene going to the father's chromosome 1. And that's practically what occurs during prophase 1 of meiosis 1. Remember that meiosis 1 is very similar to what we see in mitosis. The difference is just in the fact that we have synapses occurring, which is the pairing up of homologous chromosomes, and then there's exchange of genetic materials around um, that particular period due to the fact of the pairing of synapses that occurs prior. So moving on, the other subphases of meiosis one includes prometaphase. Prometaphase is just the transition point from the prophase to the metaphase stage. Remember that prophase one usually commences with the presence of 46 duplicated chromosomes, and from there we have the centrosome forming, the mitotic spindle also forming. We have the nuclear material disintegrating, and then the chromosomes condensing. And by the end of prophase, towards the end of prophase one, we have the mitotic spindles being completed and the synapses and exchange of genetic material occurring. Once we move from prophase one, we go into metaphase one. Metaphase one simply tends to mirror what we see in mitosis. In metaphase one, we have a homologous chromosomes aligning across the equator of the cell, and that alignment is done by way of the mitotic spindle that are formed already in prophase. So all those mitotic spindles that we saw, the cells would align themselves across the mitotic spindle around the equator, and from there at anaphase tend to lead to separation of the homologous chromosomes. So note that in anaphase it's the homologous chromosomes that are separating from one another and not necessarily the duplicated fashion of the chromosome. So because of the fact that the exchange materials, in a way, we are not even if the homologous chromosomes are being separated are around anaphase 1, in a way it's not necessarily saying that the 23 the 46 chromosomes from the mother side and the 46 chromosomes on the father side are being separated because they've already had mixtures of each other's genes. So genetic material has been mixed together. So it's usually a combined or complexly formed new chromosome that we see at anaphase, and these chromosomes tend to separate at anaphase 1. So please remember what is occurring at anaphase 1. There's disjunction or separation of paired homologous chromosomes. So it's the homologous chromosomes that are separating from one another. The one from the paternal side is separate from the one from the maternal side. But also remember that each of those different homologous chromosomes have had genetic materials being swapped for one another. Then moving on to what occurs in telophase. Telophase is the last stage of meiosis 1. So telophase 1 involves practically a reversal of what occurred during prophase 1. So we have the decondensation of the chromosomes. They begin to disappear. We have the mitotic spindle disintegrating. We have the nuclear membrane reforming, and then the cell bodies would divide at the equator to give rise to new daughter cells. So usually this occurs, and immediately after telophase 1 occurs, we have the ribosomal RNA synthesis beginning. To complete the entire meiosis 1, we have cell division finally splitting the cytoplasm of the cell into two major daughter cells. And at the end of the day, we have two cells. One of them is a diploid cell. The other is also a diploid cell. So at the end of the day, we have two daughter cells that are diploid in nature, that have a total of 2n chromosomes or 46 chromosomes. So right after meiosis 1, we move into meiosis 2. Under meiosis 2, starting with the very first phase of meiosis 2, which is prophase 2. That begins with the two daughter cells that we got from meiosis 2. Each of these two daughter cells has a total of 2n chromosomes or 46 chromosomes. These chromosomes, whereas, are not chromosomes that are homologous pairs. Remember, the homologous pairs have been separated at anaphase 1. So imagine that one of the daughter cell contains just maternal chromosomes, and these are maternal chromosomes that have mixtures of genetic materials from the paternal chromosome. And then the second genetic cell has paternal chromosomes that has mixtures of genetic material from the maternal chromosome. So that's practically important, and that's unique to remember in terms of prophase 2. So then the rest of prophase 2 will just comment or continue similarly to what happened in prophase 1. We have chromosomes condensing, and they become more visible, and the centrosome separates, and then there's formation of mitotic spindle within each of the cells. Then from there we go to the pro-metaphase stage, which is just a transition from the prophase stage to the metaphase stage. That one involves the degeneration of the nuclear membrane into small vesicles and intracellular vesicle storage, and then finally the completion of mitotic spindle formation. And the difference between prophase 1 and prophase 2 is the fact that in prophase 1 we have that synapses of homologous pairs and the crossing over of the genetic segment in prophase 1. In prophase 2 we don't have that, because in prophase 2 the homologous pairs have been separated already, so we just have a single, either a paternal or maternal set of chromosomes in each of the cells. So this is a major difference between prophase 1 and prophase 2. Once we move from the prophase stage to the metaphase stage, metaphase 2 tends to also run in a similar fashion to metaphase 1. We have each of the different chromosomes aligning themselves around the equator. So we have the total for the 6 chromosomes, what we call 23 duplicated chromosomes. Please do remember that though I have written 23 duplicated chromosomes here, they are total of 46, because 23 times 2 give us 46. So we have 46 chromosomes aligning at the equator of the cell. And at anaphase 2 usually occurs almost immediately after metaphase, after the alignment. What happens in anaphase 2 is that we now have each of these 46 chromosomes now separating. They tend to separate and to give rise to separate set of chromosomes at different poles of the cell. Now it's important to note something. For anaphase 1, when the separation of the homologous pairs of chromosome occurs, there is usually no splitting of the centromere. Centromere that tends to hold duplicated chromosomes to each other is not separated in anaphase 1. So this is practically what I mean by a duplicated chromosome. Usually a duplicated chromosome has centromere that tends to connect them together. So if this is one of the chromosome here, this is the second chromosome here. So we call them duplicated chromosomes. So we count this entire thing as one chromosome. So this is just a single chromosome, but it's actually 2 in 1, because we have one chromosome here joined to another chromosome here, which has been duplicated, just a direct copy, and that chromosome is held by a centromere. All right. So this is different from homologous pairs. Homologous pairs imagine we have another set of chromosome like this, but this one is now gotten from one of them is now a maternal, it's gotten from a maternal side, it's from the father. So this, let's say this one is a paternal chromosome, this one is a maternal chromosome. So if we have chromosome 1, let's imagine chromosome 1, imagine this is chromosome 1, and this is paternal chromosome 1, and this is maternal chromosome 1, then we say these are homologous pairs. All right, this is actually a total of 4 chromosomes. So this is 1, 2, but these are direct copies. This is 1, 2. These are direct copies. So we have a total of 4 chromosomes, but we say that each of the chromosomes there are duplicated. We have 4 chromosomes but 2 pairs. So the first pair is the paternal one, the second is the maternal, but each of them has 2 chromosomes connected by a centromere. I hope I am able to explain that very well. So if in case you're confused about the fact that I'm saying I'm typing, or I typed 23 duplicated chromosome on the slide, and I'm saying 46 chromosome, is just by virtue of the fact that one of the chromosomes is duplicated and connected together by a centromere. So do note that in anaphase 1, because we are just separating homologous pairs, centromeres are not involved in the splitting. So we just have the separation occurring there. First, the homologous pairs are not connected by centromeres. They are separate. But the reason why they appear together is that they are the same number of chromosome, but from different sources. One is paternal, the other one is maternal. But in anaphase 2, because it is a same chromosome that is duplicated but connected by the centromere, when we have anaphase 2 occurring, and that separation is occurring through the mitotic spindle, we have the centromere being destroyed. We have the splitting of the centromere. So one pair, one duplicated part of the chromosome would go to one pole, the other will go to another pole. So at the end of the day, we're having 23 chromosomes on one pole, one end, 23 on the other end. Then the rest of meiosis 2 would end with telophase. Telophase usually involves a reversal of what are called in prophase 1 and in prophase 2 rather. So in telophase 2, we have the decondensation of the chromosomes, which makes them to be invincible. We have the disintegration of the mitotic spindle, the formation of a new nuclear membrane. Cell bodies usually divide across the equatorial plane, and then finally, ribosomal RNA synthesis will begin. And then most importantly, to separate the cells completely, cell division would occur, and that will lead to the formation of not just 2 cells now, but 4 cells. Remember that from meiosis 1, we have a single cell becoming 2 cells, 2 daughter cells. Each of these daughter cells are still diploid in nature. And then after meiosis 2, we go from 2 daughter cells dividing into 4 daughter cells, and each of these daughter cells are haploid in nature. So what do I mean by diploid and haploid? Remember that at the beginning of meiosis, we started with 92 chromosomes. Each of these chromosomes are actually duplicated chromosomes, and they have homologous pairs. At the end of meiosis 1, we had a total of 46 chromosomes. All right, each of the homologous pairs have been separated. So we have a maternal one on one side and a paternal one on the other side, and they are 46 in total. Each of the 46 chromosomes are 2 in 1. So each of them are duplicated, so there are 23 duplicated chromosomes with the centromere connected to them. And at the end of meiosis 2, what occurs is that we have these 46 chromosomes now undergoing further mitotic division, and we're having 23 or haploid numbers of chromosomes in each of them. So we have 4 daughter cells with 23 chromosomes at the end. So we've talked about the very first stage of gametogenesis, and this occurs in whether we're talking about males or females, whether we're talking about the ovaries or the testes. Gametogenesis usually tends to occur first of all through meiosis, and then from there the next step would be what we call gamete maturation. So let's start with the males, starting with spermatogenesis. Spermatogenesis is the form of gametogenesis occurring in males. It occurs in the testes. It would be preceded first of all by meiosis, but after meiosis, the gametes that are formed, or the haploid cells that are formed, will now further undergo genetic maturation. So let's talk about spermatogenesis. Spermatogenesis is further divided into spermatocytogenesis and spermiogenesis. Talk about each of them in the next slide. Spermatocytogenesis is a couple of mitotic divisions that occurs in the testes, and it involves the conversion of the germ cells of the testis into that haploid cell that we talked about. Usually tends to occur in different substages. We'll talk about each of them one by one. So usually we start with a primordial germ cell. This is a germ cell that is 46 chromosomes in number, has two N chromosomes, and these cells are found within the wall of the yolk sac. At the start of embryology. They then would migrate from the yolk sac all the way to the testis. We'll talk about the yolk sac very soon in the topic of embryology week 1 and week 2. We'll make mention of the yolk sac. But in this case, the germ cells that will be found within the testis and found within the ovary usually tends to originate from the yolk sac. And before they get to the testis and the ovary, they will come from that yolk sac and migrate eventually around week 4. So for males, at puberty is when these germ cells will now start to undergo spermatogenesis. Spermatogenesis practically starts at puberty. So at puberty, we have the primordial germ cells usually tending to differentiate into two major types of cells. They differentiate into a type A spermatogonia, which is also a diploid cell. And this type A spermatogonia will now undergo further division to form a type B spermatogonia. So, like I said, type A spermatogonia usually undergoes mitotic division. So, this is important to know. So, type A spermatogonia is the very first cell derived from the primordial germ cells at the start of puberty. And this type A spermatogonia will undergo mitosis, and as they are undergoing mitosis, they are providing continuous supply of stem cells throughout reproductive life. And that's why usually in males, as a man or as a male, we don't necessarily run out of germ cells or sperm cells within our testes, and that's because of the fact that the type A spermatogonia is always replenishing the amount of stem cells found in the testes. Some of the type A spermatogonia will differentiate to become a type B spermatogonia, and this occurs through the process of mitosis. So we have type A also undergoing mitosis to form type B spermatogonia, and that would be a higher form or a more mature form of the type A spermatogonia. As we go down the process of spermatocytogenesis, we have the type B spermatogonia undergoing further maturation to form a unique set of cells called primary spermatocytes. And these are cells that contain 46 duplicated chromosomes or 4N, so they have a total of 92 chromosomes. And this mirrors what we talked about with regards to the cells that we start with during meiosis 1. So the primary spermatocytes are usually the cells that tend to enter into meiosis 1, and they undergo meiosis 1 to form the secondary spermatocyte. So we go from type A spermatogonia to type B spermatogonia, and then type B spermatogonia matures to form the primary spermatocyte. Primary spermatocyte undergoes meiosis 1 to form the secondary spermatocyte. So remember that when we are starting with meiosis 1, we have a total of 92 chromosomes, 46 duplicated chromosomes, each of them connected together by a centromere. At the end of meiosis 1, we have two daughter cells, which are the secondary spermatocytes. And secondary spermatocytes usually have a diploid set of chromosome, which is just the 46 chromosomes or 20 or the 2N chromosome. Then the secondary spermatocytes undergo meiosis 2, and they undergo meiosis 2 to give rise to the haploid set of cells we call the spermatids. So spermatids are the haploid cells that tends to form at the end of meiosis 2. So secondary spermatocytes gives rise to the spermatids. We have 4 spermatids being formed by 2 secondary spermatocytes, and each of them will contain haploid numbers of chromosomes, just 23 chromosomes, as discussed prior when talking about meiosis. So the formation of spermatids marks the end of spermatocytogenesis. The spermatids would move into the next stage of spermatogenesis, which is spermiogenesis. Spermiogenesis simply involves the process of a post-meiotic series of morphological changes by which the spermatids tend to undergo. And these changes usually involves 3 major things or 3 major processes. The first will be the formation of what we call an acrosome. Then there will be condensation of the nucleus of the spermatid, and then finally spermatid will give rise to a head, a neck, and a tail. So usually it starts to look more like a mature spermatozoa at the end of spermiogenesis. So we're having the spermatid being transformed to something unique as this shown in the image here. So yeah, so this is just a depiction of the morphological changes of the spermatid that while it's being transformed to a spermatozoa. So you can see the head there, then the neck and the tail, and at the head we have that acrosome being formed. So the head would have an acrosome formed at the tip. Acrosome contains a large amount of enzymes, and the head would also contain the nucleus that is packed together, condensed together. The neck usually tends to contain a large amount of mitochondria, and that will serve as a form of powerhouse and energy production site for the sperm cell. And then the tail contains a large amount of microtubules that involves in the propelling of the sperm cells towards this destination for fertilization to occur. The total time for sperm formation tends to be about 64 days, and that is from the process of spermatogonia formation to the process of spermatozoa formation. Newly ejaculated stem cells are usually first of all incapable of fertilization until they undergo a process we know as capacitation. So capacitation tends to occur when the sperm cells are present within the female reproductive tract, and it involves the unmasking of the sperm glycosyl transferase site and the removal of protein coating on the surface of of the sperm cell. This is very important because for the binding of the sperm cells to occur on the oocyte, which is a process in fertilization, we need those glycosyl transferase receptors to be unmasked. So capacitation is important um, for fertilization to occur, even though the sperm cells have been formed and they undergo maturation and they can swim all the way down to the uterine wall. It doesn't necessarily mean that they're capable of fertilization until capacitation occurs. So this is an image that depicts for us the entire process of spermatogenesis. Remember, we start from the primordial germ cells in the fetal structure migrating from the yolk sac to the site of the gonads, the testes in the males, and then at puberty, spermatogenesis begins by the formation or the conversion of the germ cells to a spermatogonia type A. Type A spermatogonia undergoes further mitosis to form type B spermatogonia that would undergo further maturation to form primary spermatocytes. And at this point, the primary spermatocytes will undergo meiosis 1 to form secondary spermatocytes. Secondary spermatocytes will undergo meiosis 2 to give rise to spermatids. And then spermatids undergo spermiogenesis to form spermatozoa. So moving on to the female aspect of gametogenesis, that process is known as oogenesis. It usually occurs within the ovary, and it tends to also follow a similar fashion to that of spermatogenesis, with just very specific differences. So what do I mean? First of all, we start with the process of meiosis, and then finally we have the process of maturation or gamete maturation occurring to form a full-blown oocyte. The unique differences between oogenesis and spermatogenesis is in the fact that oogenesis tends to begin before puberty and at different points in time, which we'll talk about. The process of meiosis in the process of oogenesis tends to be halted and eventually progressing at different points in time based on different scenarios or situations. So I said before, oogenesis usually begins intrauterine, and this first of all starts with the primordial germ cells migrating from the yolk sac towards the ovaries around the fourth week. Once they get to the ovaries, they don't wait until puberty occurs before they begin to differentiate. So the primordial germ cells eventually differentiate while within the uterus, while the fetus is in utero to form Eugonia. Eugonia tends to be the platelet of cells that are found within the ovaries, and this process of conversion of the primordial germ cells to the Eugonia is through mitotic diffusions. Then Eugonia will further develop or further mature to form the primary oocyte. While the fetus is still within the uterus, the primary oocyte undergoes meiosis, and this begins to occur around the fifth month of fetal life. They undergo meiosis 1 and they get arrested at the prophase stage of meiosis 1. And this arrest occurs around the fifth month of the fetal life up until puberty. So we say that oogenesis begins around the fifth month in utero. However, it's important to note that this oogenesis process is paused, is halted around the fifth month at prophase 1 up until puberty. It's after we get to puberty that we can now recommence the entire process of oogenesis in the female child. So the primary oocytes usually tend to remain arrested at the prophase stage of meiosis 1, and they become with time surrounded by follicular cells in in the ovary, these follicular cells are usually simple squamous cells, and the combination of the primary oocytes that have been arrested at prophase 1 and the follicular cells that are surrounding them is known as the primordial follicle. So usually primordial follicles are found in utero, are formed in utero, and the primordial follicles contain simple squamous cells surrounding the primary oocyte that has already been stocked at prophase 1 in meiosis 1. And we now tend to see from there, moving from the intrauterine life to birth to puberty, we tend to now see a reduction in the amount of primordial follicles from this point henceforth. So before birth, all the ogonia present within the ovaries are being converted to primary oocytes, and all those primary oocytes are stocked at the prophase 1 stage of meiosis 1. And all of these primary oocytes are also surrounded by the simple columnar epithelium, forming or giving rise to the primordial follicle. And at birth, we have a total of 1 million primordial follicles present in the ovary. And no oogonia is present there. And each of those primary follicles contain a primary oocyte that is stocked at prophase 1. From birth till puberty, the 1 million primordial follicle that we started with begins to reduce in number, it begins to degenerate. So we go from a total of 1 million primordial follicles at birth to about 400,000 primordial follicles at puberty. And it is from this stage in puberty that we now begin to see the pre-commencement of oogenesis. So usually at puberty, primary oocyte tends to first of all become surrounded by um, more epithelial cells. In this time, we have cuboidal or columnar looking follicular cells. So when we start to see the squamous cells that were surrounding the primary oocyte changing to cuboidal cells, we no longer call them primordial follicle. We now start to call them primary follicle. So at puberty, we go from a primordial follicle, which contains a primary oocyte surrounded by simple squamous cells, to a primary follicle, which contains a primary oocyte surrounded by follicular cuboidal and columnar cells. That's very important to remember. This development um, continues. The primary follicle, from being surrounded by follicular cells, now begins to be surrounded by what we call granulosa cells. So we go from just follicular cells to granulosa cells. At the point in which the primary follicle becomes surrounded by granulosa cells, we stop calling it a primary follicle and we start calling it a secondary follicle. So remember each of these different stages. So nothing is happening to the primary oocytes at puberty. Nothing yet is happening to the primary oocyte at puberty. What is practically happening to it is the fact that what is surrounding it keeps changing. So in utero, we start with um, simple squamous epithelium, and at that point we call it the primordial follicle, which is the primary oocyte and that particular structure surrounding it. At puberty, we move to a primary follicle, which is the primary oocyte that has been stopped at prophase 1 and cuboidal cells surrounding it. And then, still within puberty, this would further develop into a secondary follicle in which we have granulosa cells that are cuboidal in shape also surrounding this primary oocyte. So once the cells become a secondary follicle, the secondary follicle would further mature still within the period of puberty, usually around the very first menstrual cycle, or what we call menarche. These cells would mature to form another set of type of follicle called an antral follicle. So antral follicle is a type of a follicle that has the features of a secondary follicle, but a space forming within the center of the follicle, and that space is known as the antrum or the antral cavity. So the cavity forms between the layers of the granulosa and follicular cells, and once the antral follicle becomes obvious, the antral follicle would further develop to form what we call a Graafian follicle. So Graafian follicle is just an extension of the antral follicle. In this one, we have a third layer being added. So initially, we started with just a single layer of squamous cell, which is surrounding the primary oocyte and gives rise to the primordial follicle. Then we have that squamous cell being replaced by cuboidal cells, and in this case, cuboidal cells surrounds the primary oocyte to form the primary follicle. And then from there, these cuboidal cells are replaced by or is added on with by granulosa cells. So we have granulosa cells and the follicular cells combined together, and they are both cuboidal, surrounding the primary oocyte, and this will give rise to the secondary follicle. And then with time, secondary follicle will now develop a cavity in a space within it. That space is what we call the antrum, and at this stage, we start to call the secondary follicle an antral follicle. Then finally, a third layer is now added to the granulosa cell and the follicular cells that have been surrounding the primary oocyte. This third layer is known as theca cells, and theca cells um, plus the granulosa cells plus the follicular cells surrounding the primary oocyte with the presence of an antrum gives rise to a Graafian follicle or a mature follicle. So to put this in perspective, when a female child gets to the age of puberty, she has the very first menstrual cycle, which is what we call menarche, and that menstrual cycle practically all occurs, all the things I described from the formation of the primary follicle to the secondary follicle to the antral follicle to the Graafian follicle. So this is occurring at the very first menstrual cycle of a female child. And this tends to start from the first day all the way to the 14th day of menstruation. So as the woman, as the girl child is undergoing puberty and having a first menstrual cycle or first menstrual cycle or what we call menarche, from the first to 14th day is when all of these things are described: formation of the primary oocyte, secondary formation of the primary follicle, formation of the secondary follicle, of the antral follicle, and then maturation to the Graafian follicle. But at the 14th day, the primary oocyte buried within the Graafian follicle will complete its meiosis 1. Remember that this primary oocyte has been at meiosis 1 since intrauterine life, since when the fetus has been in the uterus, it has been stuck at the prophase stage of meiosis 1. But once we get to the 14th day, usually what we call the period of ovulation in menstrual cycle, the primary oocyte would mature and would complete meiosis 1 to form a secondary oocyte. So we only start to talk about secondary oocyte after ovulation occurs or at the period of ovulation. So we have the primary oocyte undergoing meiosis 1 to form a secondary oocyte and a first polar body. So unlike what we see for the process of spermatogenesis, where we have the primary spermatocyte the primary spermatocyte undergoing meiosis 1 to form secondary spermatocytes. And remember, we have one single primary spermatocyte forming two secondary spermatocytes. Unlike that, in oogenesis, we have a single primary oocyte undergoing meiosis 1 to form a single secondary oocyte and a polar body. So a polar body is not necessarily a cell, it's just a smaller version of a cell that is not mitosically active anymore. So that's a very unique difference between what occurs in the meiosis 1 of oogenesis and what occurs in the meiosis 1 of spermatogenesis. So for oogenesis, we go from a primary oocyte that completes meiosis 1 at ovulation, and this gives rise to a single secondary oocyte and the first polar body. The secondary oocyte will now begin to undergo meiosis 2, but similar to what we talked about for the primary oocyte, the secondary oocyte would also have its meiosis 2, the process of meiosis 2 being arrested at the metaphase stage. So remember the key difference. The primary oocyte undergoes meiosis 1. It begins to undergo meiosis 1 at the intrauterine life while the fetus is still in the uterus. And this process of meiosis 1 is altered at the prophase stage of meiosis 1. Secondary oocyte, whereas, undergoes meiosis 2. It begins to undergo meiosis 2 at ovulation or after ovulation, and not after, usually before ovulation. Let's just say before ovulation, usually around the 14th day. But the meiosis 2 become halted as well at the metaphase stage of meiosis 2. So for primary oocyte is the prophase stage of meiosis 1. For secondary oocyte is the metaphase stage of meiosis 2. And this occurs, this process occurs from ovulation till fertilization. It is only after fertilization occurs that the secondary oocyte can complete meiosis 2. So once this occurs, once the primary oocyte becomes the secondary oocyte and the secondary oocyte is crossed at metaphase stage of meiosis 2, the Graafian follicle will now release the secondary oocyte into the uterine duct. So this release of the secondary oocyte into the uterine cavity or uterine duct is known as ovulation. Ovulation is just a release of the oocyte from the Graafian follicle. So once the oocyte has been released into the uterine duct, which is the fallopian tube, the usually tends to the um, await the sperm cell to meet it and fertilize it. Usually tends to take about 24 to about 36 hours for that to occur. So whenever there's ovulation, there's a space of about 2 days for a woman to become to undergo conception and to be pregnant at the end of the day. So the oocyte that is released usually tends to just hang around the uterine duct, hang around the fallopian tube. And if fertilization does occur, if that oocyte comes in contact with a sperm cell and the sperm cell starts to fertilize it, the secondary oocyte that has been stuck at meiosis 2 since ovulation would undergo completion of that meiosis 2. And once it completes meiosis 2, it becomes a mature oocyte. So secondary oocyte becomes a mature oocyte and it forms a second polar body. So this is also unlike what we see from meiosis 2 in terms of the process of spermatogenesis. In spermatogenesis, we in meiosis 2 we start with two secondary oocytes, and these two secondary oocytes would eventually undergo meiosis 2 to form four spermatids. So we go from 2 to 4, and that's what occurs for spermatogenesis. Contrast that to oogenesis. In oogenesis, we have a single secondary oocyte undergoing meiosis 2 to form a single mature oocyte, or what we call an ovum, plus a polar body. So it's always a single primary oocyte forming a single secondary oocyte plus the polar body, and that's the first polar body. Now we go from a secondary oocyte, single secondary oocyte forming a mature oocyte or mature ovum plus a second polar body. And this is the image that depicts for us all the things we talked about in terms of the development of uh, oogenesis. We start with the gonium. Gonium is usually the two-end or diploid set of cells that undergoes mitotic division to form primary oocyte. Primary oocyte to begin meiosis 1 and is arrested at the prophyte stage of meiosis 1 as shown in image here. This is only completed when we get to puberty. At puberty, at around the 14th day of the menstrual cycle, the primary oocyte stuck at meiosis 1 would complete that meiosis 1 to become a secondary oocyte. Secondary oocyte commences meiosis 2 but becomes arrested at the metaphase stage of meiosis 2. And this remains until that secondary oocyte becomes fertilized by the sperm cell. At fertilization, we have the secondary oocyte completing meiosis 2 to give rise to a fully mature ovum. Now, contrast the process of oogenesis to what occurs in line with oogenesis, which is the formation of follicles. Remember that we talked about the fact that as we go from the intrauterine life to puberty to birth to puberty and all the way to ovulation, aside the fact that we have an oocyte present within the ovary, we also have follicles developing around them. So in terms of follicular development, we start with primordial follicle, which is at intrauterine life, to a primary follicle, which is at ovulation, and which is at puberty. Then that primary follicle matures to a secondary follicle that eventually becomes an anterior follicle, and then finally it becomes a mature follicle, Graafian follicle. Then at ovulation, the mature follicle would rupture and release the secondary oocyte into the uterine duct. And then from there, that will lead to the formation, or that would eventually lead to the formation of the corpus luteum. The corpus luteum is just the remnants of the follicles, all those granulosa cell, follicular cell, and thicker cells that have been forming around the oocyte. Once they've released the oocyte, they will become what we call the corpus luteum. Now, this is just showing us the development of the follicle in terms or alongside what occurs in oogenesis. In oogenesis, it's a different thing entirely. You are focusing only on the cell, only on the germ cell. We start with the germ cell that becomes gonium. Gonium becomes primary oocyte. Primary oocyte is arrested till puberty at prophase 1, and then at puberty, the primary oocyte would, before ovulation, undergo maturation of meiosis 1 to form secondary oocyte. Secondary oocyte begins meiosis 2 but becomes stopped or arrested at the metaphase stage of meiosis 2, and then eventually at fertilization, that secondary oocyte be would be able to complete its meiosis two and form a mature oocyte. Mature oocyte there would now further undergo fertilization to form the zygotes, which is what we'll talk about in the um, slide under fertilization. That is on embryology week one. And that completes our module on gametogenesis.